Okay. Yeah. Okay.
No,
Yeah.
I sometimes brush it, but I sometimes even pause it for it on.
Okay, so we are going to continue talking about digestion today. So we're going to finish up looking at our accessory glands by looking at the liver. Then we're going to move into talking about the digestion and absorption of our different macromolecule types one by one. Okay, so eating a snack, this is going on right now. If not, think about it very much. Okay, so as we were talking about our accessory glands last time, we talked about our salivary glands producing saliva, beginning digestion in the mouth. We had looked at the pieces of the GI tract, so the tubes that your food is moving through. We had looked at the pancreas, secreting some pancreatic enzymes. We had seen that some enzymes are coming from the walls of the stomach actually as well. So part of our GI tract secreting some enzymes. So now we're talking about the liver. We've seen the liver before once or twice. So we've looked at it in processing red blood cells. We've mentioned clotting factors coming from it. Now we're talking about its digestive properties. You've seen this in lab, actually, the structure of it. Okay, so the liver, its digestive process is to secrete bile. So the bile is going to help us process nutrients. We've seen some of the structures that bile travels through. So in lab, you probably saw, dyed bright green by actual bile, not by a dye, that we have our hepatic duct coming out of the liver, joining up with the cystic duct from the gallbladder, joining together to form our common bile duct, which is then going into the duodenum at the sphincter of Oddi. At the same point, we're actually going to have the pancreatic duct joining in as well. So we're going to get all those good enzymes and bile salts moving into the start of our small intestine so that we can break down all these compounds and absorb them across the walls of the GI tract. Okay. So the liver is secreting bile. Some other jobs of the liver: processing nutrients. So we saw that glycogen is a storage molecule for glucose. We mentioned it back in A&P 1, that this was one of our sort of sources for storing glucose and then using it or having glucose to use to produce ATP when we haven't just eaten a meal. So one of the places that storage of glucose as glycogen happens is in the liver. Also can turn amino acids into fatty acids, synthesizes triglycerides and cholesterol, synthesizes lipoproteins, so combination fat protein molecules, okay. Removes old blood cells. What it's doing in that process is catabolizing, so breaking down hemoglobin, generates bilirubin as its byproduct. It's helping us eliminate waste and toxins like alcohol. Bilirubin is actually what gives color to your feces, makes it brown. So you saw that bright green bile, bilirubin has color. Also synthesizing plasma proteins like those clotting factors. And it secretes, modifies hormones. The liver does tons and tons of stuff. Okay. All right, but we're thinking about digestion. So we're really thinking mainly about the biliary system and those functions of the liver in this unit. Okay, so the biliary system consists of the structures related to bile. So the liver, the gallbladder, which is really just a storage organ for our bile, as well as the ducts associated with these organs. So the liver is actually making the bile. And we'll take a look at some schematics of liver cells, sort of internal liver structure, let's say. So the liver is making the bile. The bile goes through that hepatic duct, that common hepatic duct. And if you're not using that bile immediately, we have spillover and we travel into the gallbladder and store that bile for later. So that's what the gallbladder is for. That's why you can remove the gallbladder and be kind of fine, right? So bile stored here in the gallbladder. These tubes are connected in the common bile duct. So when you're ready to digest a meal, particularly a fatty meal, you're going to spill that bile into the common bile duct, send it into the duodenum. Okay. At that point, we're joining with the pancreatic duct. So we're going to get other good enzymes coming from the pancreas here, helping us digest other components of our food that we'll talk about. This sort of swelling where they're joining is called the ampulla of Vater. Ampulla, I think, is some old Greek or Roman type of like jug. So anything that looks swollen is often called an ampulla. That's just a note about anatomy terms and where they come from. So ampulla of Vater, and then the sphincter of Oddi is kind of controlling the release of these enzymes into the duodenum. So looking inside the liver has lobes. You can donate pieces of your liver and be fine. It's kind of easy. I don't know how easy it is with your hands in terms of surgery, but for your processes, like you're doing fine in life after, if they remove part of your liver, you're fine. Easy to remove different parts. Okay. So if we look into the liver, we have different lobes of the liver. And we have surgically resectable, so removable lobes of the liver in addition to anatomical lobes. But if we were to look inside those lobes, the liver also has little lobules where it's producing bile and having exchange going on between its blood. So we're looking at those little lobules here, these little hexagons. Okay, so we can see that in each of these hexagons, we have a central vein, right, colored in blue here in the center. That sort of point in the middle of our little flower star thing, okay? And along the edges, we have little triads consisting of a vein, an artery to feed that tissue, and a little bitty duct, okay? So we're getting blood flow, we're getting blood removable, and we have somewhere to put the bile that we're producing. Okay. So the cells of the liver are called hepatocytes. So hepato, anytime you see that root, that's talking about the liver. Cytes just tells you that's a cell. Okay, so the hepatocytes are making bile, and I don't think we have bile canaliculi labeled on here. So if we were to zoom in even further, you'd see that we have canaliculi, think like canal. So we'd have like little canals in here. So the cells would secrete bile into these little canals, these canaliculi, and then drain into the biliary system. Sinusoids are kind of the same thing. Also, I think not drawn on here, but that's where we'd have blood and products exchanging across it. So that would, again, just be like a little internal thing. So sinusoids, think like a sine wave. So it's going to be something kind of like curvy. Okay. So that's all that we're looking at when we're thinking about bile production. We have these cells producing bile, sending it into our biliary system. Okay. So here we have just a table from your book about the organs in our gastrointestinal system and what they're doing. But we're going to go ahead and move straight into absorption so that we can look at this from more of a biochemistry perspective. I don't know if it's fair to call this biochemistry, but it's biology and it's chemistry, so I'm going to go with it. So we're going to be talking about the digestion of carbohydrates, proteins, and lipids. All right. So when you eat food, you need to get all of these components into your body, into your cells, so that you can do our cell processes, right? We need to get carbohydrates small enough that we can absorb them across our cell, get them into the plasma, preferably in the form of glucose, so we can ship that around to cells that are actively making ATP through metabolism. We need proteins and amino acids so that we can build all sorts of cellular machinery, as well as building muscle cells, all sorts of good stuff like that. Enzymes, basically all sorts of things in your body are made of proteins. I can't list them all off the top of my head. We need some lipids, great alternate source of energy, as well as sort of storage molecules. Okay. We also need various vitamins and minerals that are involved in some of the chemicals that your body makes, some of the structures that your body makes. We especially need like salts, right? So we need ions and we need lots of water. We want to keep up our hydration levels. We want to keep up our plasma volume. So all of this is going to be coming from your food, and we have to get it across the walls of the GI tract. We have to get it small enough, essentially, that a cell can take it in, right? Otherwise, it's just going to stay in your GI tract and get excreted out as feces if it's too big. Okay, so we're going to go through our carbs, our proteins, and our lipids and how they get into our body. Okay, so say we're eating a sandwich, right? So we have some bread, we got a carbohydrate, so we want to get that into our body. As we start to digest that, right, first you chew your sandwich, right? So you're mechanically breaking it down first. That's mastication. Okay. And carbohydrate digestion is actually going to begin in the mouth. That's because our salivary glands, remember, we're producing saliva. One of the components of that saliva was our salivary amylase. So salivary amylase begins breaking up like our starch molecules, essentially. So here we see a picture at the top of starch. So starch is like a plant storage molecule for glucose. So each of these little rings is a glucose. You can see that we have all sorts of branches that we could ultimately cut apart to get out those glucose monomers, right? And we've talked about glucose transport before. We know glucose can get into cells, co-transported with sodium, right? We've seen where that happens. For example, as we were talking about reabsorption of glucose in the kidney. Okay, so your cells are only going to really be able to absorb monosaccharides, so little individual units. Glucose is not our only monosaccharide. It's just the one we focus most on because we care most about, easiest to process for us. Okay, so we got to break up this starch molecule in order to get it into our cells, into our plasma. Okay, so this is a big polysaccharide, right? We couldn't even get in so much as really a disaccharide across a cell, still kind of too big, and we don't have the right carrier proteins for it. Right, so in the mouth, our salivary amylase is going to start chopping up this molecule, essentially. So we're going to start chopping. All right, so you're going to begin that process of digestion. You're going to swallow your bolus of food. It's going to go down to your stomach, get mixed up with stomach acids. Then it's going to get squeezed through the pyloric sphincter into the duodenum, mixed in with everything in that chyme. That's sort of like digestion smoothie that's going into your duodenum, right? That's where it's going to encounter pancreatic amylase, which is our next enzyme that breaks up carbohydrates, okay? So salivary amylase in your mouth and pancreatic amylase that's coming from that pancreatic duct that we just saw joining up with our biliary system, going through that sphincter of Oddi into the duodenum. Both of these are going to help us break down our big polysaccharides, for example, starch. Okay, so they're breaking up the starch. They're cleaving it like a cleaver, breaking it apart. But they actually can't fully get us into individual glucose monomers. That's just not how they work. Okay, so what those two are going to leave us with are disaccharides and limit dextrins. Limit dextrins are just kind of like these corners. The corners are specifically kind of hard to get at with our enzymes that we have with those amylases. So this digestion process would continue with other enzymes that are kind of embedded on the walls of cells in our GI tract that would ultimately break it down to monomers, so like individual glucose units, which could then be transported into the cells. For example, if we have a glucose, can get co-transported with sodium into a cell. Okay. So digesting this, most important molecules are our salivary amylase and our pancreatic amylase. They only get us so far. They don't get us quite all the way there. We have other enzymes that we're not going to name, depending on specifically what glucose or what saccharides we're talking about that help us split these up a little bit more so that we can pull them into cells. Okay. So next thing, we're going to talk about our proteins. Say we're eating, I don't know, a turkey sandwich. Okay, we want to digest our turkey. Okay. So the proteins there are going to be broken down by molecules called proteases, right? So protein for the protein, right? Proteases, ACE telling us it's an enzyme that breaks something down, okay? So we have different classes or groups of proteases. They do different things. They do different jobs a little bit. Okay, so we have our endopeptidases, we have our exopeptidases, and we have our zymogens. Okay. So these are roughly divided based on what type of bonds they're breaking. Okay. So if we think about the structure of a polypeptide, right, we have like a beaded chain of amino acids, kind of. Okay. So these are my amino acids. This is my polypeptide chain. So when you ate your turkey sandwich, you chewed up the sandwich, began that mechanical digestion. Don't have any of these proteases that we're interested in really beginning in the mouth. But once we get to the stomach, we know that we're going to start denaturing proteins with that acidic environment. Right? That acidic environment is part of what's helping us unfold a big foldy protein so that we can have a nice little polypeptide chain like this. And we know that we had pepsinogen secreted from some of those cells in the gastric pits, which is going to be important as we look at some of the processes for protein digestion. But it's that denaturing, that unfolding in the stomach that allows us to have this polypeptide. Okay, so once we have our polypeptide, I'm going to make mine a little bit longer actually, so that we can emphasize this difference. Okay, so our endopeptidases, endo, think inside. So they're just going to help us get to these inside bonds like there or there, right? Exopeptidases, exo, think outside. They're going to be able to pull off things from the end, right? So we have our endopeptidases chopping in the middle, exopeptidases pulling off amino acids from the end. And then we have our zymogens, okay? So like when I was talking about the stomach, right? We didn't want to digest the stomach tissue itself. We also don't want to digest our small intestine itself. So a bunch of our proteases are stored in inactive form that then need to be activated in order to break apart proteins. So that's what our zymogens are. And in fact, we've been looking at that sort of ogen ending on a lot of our proteins, right? And noting that that's kind of our indicator that this might be an inactive form of like a clotting factor or our pepsinogen when we're looking at the stomach. Right, so zymogens are these inactive storage forms of our proteases, so our enzymes that break up proteins. Okay, so these zymogens are stored in zymogen granules inside a cell. They then get secreted out of the cell by exocytosis. So think about like analogous to the process of secreting our neurotransmitters or something, right? So instead of a vesicle, we have a zymogen granule inside our cell, but it would bubble out to the edge of the cell membrane and then get released outside the cell by exocytosis in the same kind of way. Okay. So then once we have a zymogen floating around in, say, our GI tract, we're going to need to activate it. We activate it by proteolysis. So proteolysis, so breaking up a protein. So these zymogens themselves are proteins because all our enzymes are proteins, right? An enzyme is a protein. Okay. So we're going to look some more at this process. So that protein digestion is starting in the stomach. So we're looking at that pepsin again, right? So we had an inactive form being secreted in the stomach. So we had that pepsinogen, right, coming out of our chief cells here in the gastric pits. So it's secreted into the lumen, so the space inside our stomach. Okay. And on the other side of the gastric pit, or from the other cells in the gastric pit, we were secreting hydrogen ions, really hydrochloric acid here into the lumen of the stomach as well. Okay. So that pepsinogen, right, that inactive form, then gets activated by contact with that acid, okay, forming pepsin, which is now an active enzyme that is able to start cleaving our molecules, so to start breaking apart our proteins into smaller fragments, okay? So we can here see the pepsin coming up to that protein, that unfolded protein, and starting to chop it up, all right? So based on what these smaller peptide fragments look like, we might expect that pepsin acts kind of as an endopeptidase, right? We're not pulling just individual things off the end. It looks like we've kind of chopped them up. Okay. So that's the basic idea of how this would work. And that's all we have to worry about going on in the stomach. But once we get to the small intestine, we have a bunch more enzymes to think about. Okay. So most of our proteases, or at least more of our proteases, are coming from our pancreas. So that's a big portion of our pancreatic juices coming down through our pancreatic duct. Okay. So our pancreas is secreting these proteases, these enzymes that are going to help protein digestion. So we draw our pancreas somewhere over to the right here, right? This would be pancreas. Okay, so we're looking at the pancreatic duct. We have that pancreatic juice coming in, right? So this would be kind of going through our sphincter of Oddi, we would think about it. And three of them we're going to have in there are ultimately going to be trypsin, chymotrypsin, and carboxypeptidase. But we can see that they are beginning in zymogen form, so in inactive form. So we can see here that trypsinogen, chymotrypsinogen, and procarboxypeptidase. Okay. So these are coming from the pancreatic duct. And coming down from up here, right, we have our chyme coming from the stomach. So all that swirled together, their food and gastric juices that is partially digested because we had the pepsin working on breaking up some of our proteins in the stomach. Okay. So this digestive process is going to continue. So you can see that we activate those zymogens. So we can see trypsinogen here being activated into trypsin, chymotrypsinogen turning into chymotrypsin, procarboxypeptidase turning into carboxypeptidase. Okay. Now, some of this activation is happening because of contact with other enzymes. Okay. So we also have some brush border proteases, and we have on this diagram, one of them sort of shown here. So you can see this enterokinase is kind of on top of this cell wall, right? So this is, the cell membrane, right? So this edge of the lumen, the border here, it's stuck there, right? It's not just drawn there, it's part of it. So the brush border was briefly mentioned previously. So when we were talking about how we expand surface area in the GI tract in order to allow a lot of room for absorption, we talked about the fact that we had these projections called villi, so big ones, and then we had on our individual cells, our microvilli, which is why we draw apical membranes kind of like that, right? Because we're reminding ourselves that we have these microvilli, and then a bunch of microvilli next to each other on different cells. Right, we would refer to this as the brush border because it looks like a brush. So when we say it's a brush border protease, we're saying that we have this protease, so this enzyme that can break up proteins, like embedded here in that brush border on these cells. So that's what they're showing here with the enterokinase. Okay? So enterokinase here we can see is an enzyme that breaks up a protein, but the protein that it's breaking up here is our zymogen trypsinogen, so our inactive form. So our enzymes are proteins. So our zymogens, our inactive forms of enzymes, are also proteins. So the way that we have activated here the trypsinogen is by contacting this brush border protease. So this enzyme in the wall of the GI tract, it broke up that trypsinogen a little bit and turned it into trypsin. So that's like a concrete example of what the previous slide was saying here about the proteolysis, right? That's what we're talking about when we say that. All right. So we are continuing our protein digestion in the small intestine after we activate these zymogens in our pancreatic juices, have them in their active form as they come into contact with some brush border proteases that activate them. Right. So we are not fully done yet because we need to absorb those amino acids. And part of the reason there are so many of those proteases instead of just like we only had to remember really the word amylase when we're thinking about that starch digestion, is there are a lot of different amino acids. So we need kind of different enzymes to break up different bonds between different amino acids. Okay. But now we want to absorb those amino acids. Okay, so this is going to work kind of like our glucose transport did essentially. So before we saw that we make ATP using our sodium potassium pump, right? That's what's up here. That set up a sodium gradient for us that allowed for glucose transport with sodium, right? So we're going to have a similar thing happening with our amino acids. I don't have a picture of that, but it would work in the same way. So whenever you see sodium linked, right? So we're thinking about the fact that we set up these sodium gradients. That means sodium wants to go in a specific direction, and it can pull along other molecules by secondary active transport. So active, because we needed to use ATP to make that sodium gradient with the pump. Secondary, because we're like pulling a second molecule along with it. So it's kind of coming along for the ride, taking advantage of that earlier use of ATP. Yeah. So many of our amino acids are crossing the membrane through secondary active transport linked to this sodium. Depending on the amino acids, some will be going through facilitated diffusion. So that was when we had like a channel or something in the membrane, allowing things to go through. On the basolateral membrane, they're just going to go through facilitated diffusion. So we're not going to have to link that up to sodium. We're then going to have a favorable gradient for that amino acid that it can flow down without any energy. For dipeptides and tripeptides, so say we haven't fully broken down our polypeptide train, we'd left maybe 2 or 3, right? So we have a dipeptide or we have a tripeptide, depending on how many amino acids are linked together. So some of these we can pull into our cells. So that's a little different than the carbohydrates. In the carbohydrates, we just wanted to pull in monomers, so individual units, right, like glucose. Dipeptides and tripeptides, so 2 amino acids or 3 amino acids linked together, can sometimes cross by active transport into our cells. But inside the cell, they would then get broken down to individual amino acids and continue to cross across then as their individual amino acids into the blood. And the basolateral membrane is just the side of the cell, remember, that's always facing the blood. That's how we define it. Okay. Moving on to our lipids. So say you got butter or mayonnaise or something on your turkey sandwich, oil, I don't know what you put in there, right? We got some type of fat here on our sandwich. Maybe it has avocado, who knows? All right, so your typical diet has about 50 grams per day of lipids. 90% of that is in the form of triglycerides. So remember, our triglycerides look kind of like a flag, right? So we have our glycerol black backbone. That's where the glyc part comes from. And then 3 tri is for our fatty acid chains, right? So that's our triglycerides. Those are our fats and oils, right? Other lipids included like steroids, like cholesterol, for example. Yeah, we had a bunch of different types of lipids. They're really just all united by the fact that they are non-polar, so just like hydrocarbon chains mainly. Okay. So lipids have a problem with digestion and absorption for the reason that they are non-polar. They're just these hydrocarbon chains mainly, which means that they are hydrophobic, which means that they're not water soluble. Most of the environment in your body is water, right? So as you're swallowing, there's some liquid in there. They're joining with liquid in the stomach. We got water all over the place. So the lipids really don't mix well with the contents of your stomach or the contents of your intestine because of all the water that's in all the fluids in these organs, okay? So if you think about going through our GI tract or the stomach, I'm going to draw GI tract, okay? Usually red is blood. Here it's going to be GI tract, right? So you have lots of water hanging out here as we're going through, like in the chyme, right? That mixed together, chopped up food and water and gastric juices, right? So any fats, right? They're not going to stay nice and separate because they're trying to escape the water. So they're going to start to all kind of glob together, right? Because they're just trying to escape water because they're hydrophobic, because they're non-polar. Okay? So lipids form fat droplets as they travel through your body because of this chemical property. Yeah. So we're going to have to do special things in order to break up those fat droplets. Okay. So we're going to be using 2 things. We are still going to need an enzyme. So lipases, right, are going to break down our lipids. So lip for lipid, ACE telling us we're breaking it down. And those are coming from the pancreas. But they can't get into the middle of a big fat droplet, right? They're only going to be able to act on molecules that they're contacting. So we're also going to need to break up those fat droplets, make them smaller, make lots of surface area, okay? So we don't want to just break down the fats over here. So this little zigzag I'm drawing, say that's a lipase, right? Then we'd only be able to break down this little part. We want to be able to break down everything. So we got to break that big fat globule down. Okay. So that's what the bile salts are doing. So those bile salts, that bile coming from the liver, are going to help us break down those big fat droplets into smaller little ones that are therefore going to have more surface area, expose more of these lipids to the lipases. So they're going to expose your lipids to the enzymes that can actually break them down. And that's going to help us digest stuff better. Okay, so this process is called emulsification, this breaking down, because those bile salts are going to be able to interact well with the water, and they can also interact with the lipid, because that's how they're going to be able to break down the larger globule. Okay, so we're going to look at that. All right, so the bile salts are being made by the liver. Okay, so they are derived from cholesterol. So you can see that little 4-ring steroid bit, right? So that's typical of our cholesterol. And then we just modified it into our bile salt. Okay, so this is our bile salt made in the liver. Okay, it's coming down through our hepatic duct into our common bile duct, joining up with our pancreatic duct, going out through our sphincter of Oddi into the duodenum, okay? So our bile salts are amphipathic molecules. So we've seen that word a long time ago, right? To describe our phospholipids, right? So they're similar in this way to the phospholipids that make up the cell membrane. So amphipathic, meaning that they have a polar part that is hydrophilic, so likes water, and they have a non-polar part that is hydrophobic, which is therefore able to interact with our fats, with our lipids, okay? So that's how they can emulsify our fats, because they can interact with the water in your GI tract, and they can also interact with our bigger kind of fat droplet, okay? So here we see some hydrophilic groups, and there we see some hydrophobic parts. So we have our big fat globule, which is nonpolar. We got our water, which is polar. We have our bile salts. So we can see they have both polar and non-polar parts, which means that they can arrange themselves on our fat droplet with their polar parts facing the watery environment and their non-polar parts facing our nice little fat droplet. Okay, so they're going to help everything kind of mix together better, break this apart. They kind of surround and protect our smaller fat droplets. So instead of having a big fat globule, now we got littler fat droplets here. Okay. So most of what's in these fat droplets are things like our triglycerides, right? That was 90% of your diet. We might have some phospholipids, right, from cell membranes. Okay. So these are contents of our fat globule, just like anything that's nonpolar, anything that's a lipid is in here. Okay. So when we digest, specifically our triglycerides is what we're going to focus on and care about, because that's 90% of the lipids you're consuming. Right. We're using our pancreatic lipases. So pancreatic because they're coming from the pancreas, and lipases because we're breaking down a lipid. I'm going to draw us out a bigger triglyceride. Okay. So they are going to help us break off essentially fatty acid chains from this to make this molecule smaller. So we're going to try and get like individual fatty acid chains off of this. Okay. So we're going to break these into different parts. So a monoglyceride and then 3 fatty acid chains, hopefully. So some fatty acids and monoglycerides can be absorbed, right? So some of our lipids we are absorbing. Others are going to form a small structure called a micelle. We'll take a look at a picture, but a micelle is like where we have little globules kind of protecting themselves like this. The picture will be better. Okay. And there's kind of an equilibrium between micelles and the fatty acids and monoglycerides that are formed in the action of the lipases. So we'll look at what that means. Okay. So let's take a look at this picture. All right. So here we have our fat droplet, right? So our fat droplet, this yellow thing. Here we have our bile salt attached to the outside of it so that it can mix in better with the water. So it's been emulsified. We made it smaller. Okay? And we can see that within it, it's got some triglycerides. It's got some phospholipids. All right. It's got a bunch of stuff kind of jammed together. Okay. So the next thing that happens, right, is we secrete some lipases from the pancreas. So that's what we're looking at here with this little enzyme here. The lipase is going to help us break apart. Our triglyceride is what we're going to focus on understanding. So you can see it grabbing on to one of our triglyceride molecules. So triglycerides are the ones with 3 fatty acid tails. So you can see how it's grabbed on kind of like a cap here on this one on this image. Okay. So it can then break that up. And the way it does that is it's basically going to be chopping off a fatty acid here, chopping off a fatty acid here, and kind of leaving this middle one attached to the glycerol backbone. Okay. So we can see that then floating around in the environment in our GI tract here. So now we have individual fatty acids. Okay. So let's circle 2 of them because we're just going to track this one triglyceride that I circled in red here. Okay. So 2 fatty acids. And here we have the third one still attached to the glycerol backbone. So this little kind of T-shaped thing is our monoglyceride. Mono, because we have one fatty acid left on it. Glyceride, because of that glycerol backbone from the triglyceride. Okay. So these are hanging out here. They're still lipids. They still don't like water, right? So a bunch of them are going to form these structures called micelles. So the micelle is this little bubble here. So it's smaller than our fat droplet. Now we have like individual pieces of molecules, smaller. So we can see that we have our fatty acid chains, those hydrophobic pieces, those hydrocarbons facing in towards the center. So they're just touching other hydrophobic stuff. And then we can see that there were some phospholipids involved in this one with their polar heads sticking out of the top. We got little ends of our fatty acid chains. We got the glycerol backbone kind of on the outside touching the water. Okay. So if you ever use like micellar water or something to clean your face or something, what you're talking about is little oil droplets essentially suspended in water, right? That's what a micelle is. So we have these micelles from our partially digested, well, really our digested triglycerides. So I'm considering them digested at this point because they're broken apart, but they haven't yet been absorbed into the body. Okay. So the absorption has to happen across our epithelial lining. So across the wall of the GI tract. So you can see that some of them are just going across, right? And some of them are hanging out still as micelles in the lumen of the GI tract. And there's a balance between these. So that's really all we mean by an equilibrium. Is that basically if we have more fatty acids hanging out here, right, more of them are going to form micelles, and then more of them are going to go into the absorptive cells. Okay. So unlike our carbohydrates and our proteins, because of this different chemical property, because they're nonpolar, because they're hydrophobic, we don't actually need a transporter or anything, no proteins, no channels, nothing to get these into our cell, right? You can see this arrow, right? Like it really is just going straight across here. Okay, so they're going via simple diffusion. So our other molecules we saw, our proteins, were sometimes traveling by facilitated diffusion, like they're carrier transporter, but in a favorable direction. But these ones are going by simple diffusion, meaning they're just like straight up going directly through our phospholipid bilayer. And they can do that because they're nonpolar, because they're hydrophobic, because they're small. Okay. So that different chemical property makes a difference for how they're absorbed into the cell. So once they're in the cell, we're not totally done yet. Okay. So we have now monoglycerides. We have these fatty acid chains in an absorptive cell. Okay. Inside that epithelial cell, they're going to go into your smooth endoplasmic reticulum, and we're going to rebuild that triglyceride, right? It was too big actually to just get across our cell membrane, but the smooth ER is going to reform some triglycerides or potentially assemble them into some other lipid. And then those reformed triglycerides are going to go to the Golgi apparatus, right? So another organelle in our cell. Think way back to like your prerequisite courses for organelles. And we're going to package these into something called a chylomicron. So chylomicron is like our storage sort of form right now for our absorptive cell for these lipids. So the chylomicrons then are going to be these big bundles, essentially, of these lipids. And that is what is going to get secreted into our interstitial fluid. So this would be a chylomicron. Chylomicron. Okay. All right. This is now big again, so it can't just diffuse across the membrane. It's going to have to enter through exocytosis. Right, so we'd imagine it getting closer to the membrane and then merging to the membrane and exploding and sending its stuff out, essentially. So those chylomicrons are going to go into the interstitial fluid And then they're going to enter your lymphatic system. So that's another difference from our carbs and our proteins. Our carbohydrates and our proteins, they're water soluble. They're going through embedded proteins in our cell membranes and they're entering the plasma, so entering the blood. But our lipids are going into our lymphatic system, and they're going to have to travel through that lymphatic system before they ultimately get into circulation. So they're doing a bunch of different stuff. So here we have a picture of what's going on in that absorptive cell as well. So we had our digested products of our triglyceride in the lumen. They diffused across the cell membrane, which they can do because they're hydrophobic and nonpolar. We pulled those pieces into our smooth ER and rebuilt our triglyceride. Went to the Golgi apparatus where it mixed it up with other lipids and proteins, built a chylomicron, which is then going into our interstitial fluid, bypassing the plasma and the cardiovascular system and going into our lymphatic system. So when we were looking at our villi, not the microvilli, right, but the larger projections that were made up of many cells, right? Right, imagine this made up of lots of cells. For time, I'm not going to draw them all. Right, within each of those villi, we mentioned that we had blood flow, right? So we had an arteriole going in, we had a venule coming out, we had capillaries between them, but we also had this little branch of our lymphatic system going in. So that's the lacteal. So that's what's labeled here. So this is pointing out that these lipids, instead of going into the capillaries and getting exchanged with our cardiovascular system, they're going into our lymphatic system. Okay. And our lymph does eventually deposit its fluids into the veins. And so they do eventually go into general circulation, but they're going to travel through like these lymphatic veins and stuff first before we get them into our body. So fat digestion and fat absorption are pretty different from digestion and absorption of our other molecules. So here we just see a picture of some bile salts moving around. I think the point of this is just to show us that we are going to reuse those bile salts. I think that's what this is about. So we can see those bile salts then going back into our liver ultimately. Cool. So we're going to end there. We'll talk about briefly absorption of other things and then move into motility next week.